Welcome back. In the previous video, we have discussed about the different types of tissues seen in plants. In general, plants have meristematic tissues which result in the growth of the plant. These meristematic tissues they undergo differentiation and get converted into simple permanent and complex permanent tissues. If you look at the plant body as a whole single unit, then we have to figure out the different types of tissues where they are present, the exact location of the different tissues in the plant. In general, the plant body, we can say that it has three types of tissue systems. The entire plant body present in the root, stem as well as in the leaf. The three types of tissue systems are: number one, we have the epidermal tissue system, then we have the ground tissue, or it's also called as the fundamental tissue system, and we have the vascular tissue system. So these three are the different types of tissue systems seen in plants. Let's discuss about them one by one. First, we have the epidermal tissue system. The epidermal tissue system, it consists of the epidermis, the stomata and the hairs or the trichome. So together this consists of the epidermal tissue system. Epidermis, it is the outermost layer in a plant, in a root or in a stem or in a leaf. The outermost layer is the epidermal layer. It is generally single layer thick, so single celled thick, and the cells are compactly present with no intercellular space. The main function of the epidermal tissue system is to offer protection and also acts as a defensive barrier. So that is about the epidermal layer, epidermis. In the epidermis layer, we have what is called as the stomata. The stomata, it consists of two kidney-shaped cells called as the guard cells. So in dicot it is kidney shaped, whereas in monocot it is dumbbell shaped. So there are two guard cells which are present. So these are kidney shaped in dicot. These guard cells, they have chloroplasts present in them, so they can perform photosynthesis. The guard cells are surrounded by epidermal cells. Sometimes some epidermal cells they become specialized and they also become the subsidiary cells. And we have the guard cells. The two guard cells, they enclose a small opening called as the stomatal opening or the stomatal pore. This stomata, it plays an important role. Number one, in the process of transpiration, that is losing extra water, and also this stomata is the organ through which exchange of gases takes place. So plants, they obtain carbon dioxide through this stomata, and oxygen generated in the process of photosynthesis is thrown out. Similarly, excessive water is also lost through this stomatal opening or stomatal pore. The opening and closing of the stomatal opening of the opening and closing of the stomata is controlled by the guard cell. So when water enters into the guard cells will swell, and the pore and the stomatal pore will open. When the guard cells they lose water, it will shrink, thus closing the stomatal pore. So opening and closing of the stomata is controlled by the guard cell. This is the main function of the stomata. Sometimes the epidermal layer, the epidermis, it is covered by a outer waxy layer called as the cuticle. The cuticle, it once again plays an important role in controlling the loss of water. It also acts as a defensive barrier. It will protect the plant from microbial attack, etc. So the outermost layer we can see is the waxy cuticle. Then we have the epidermal cells, one cell thick, and we have the stomata seen in the epidermal tissue system. And we also have the hairs and the trichomes. The hairs and trichomes, hairs are usually seen in the roots, whereas trichomes are seen in the stem or in the shoot system. Hairs are unicellular, whereas the trichome, it is multicellular. So what we see, the presence of root hairs. So these are unicellular cells that are seen, which is single celled thick. The trichome, it could be either branched or unbranched. It could be either smooth or it could be stiff and hard. So the root hairs, they play an important role once again in prevention of transpiration, and the root hairs, they play an important role in absorption of water, whereas trichomes, they play an important role in preventing transpiration. So it will control the excessive loss of water. The presence of trichomes, it will discourage the animals from eating these plants. So they once again form protective barrier. Together, this is the epidermal tissue system, which is the outermost covering of the root, the stem or the leaf, made up of the epidermis layer, the stomata, and the unicellular hairs or the multicellular trichomes. The second type of tissue system what we have, that is the ground tissue system or the fundamental tissue system. The ground tissue system or fundamental tissue system, as the name suggests, this type of tissue system is present as a major part of all the plants. So they are seen in the stem, leaf and roots. They act as filling tissues, maybe providing mechanical support, strength, or acting as storage tissues. So these are present in major part of the stem, leaf and root. The ground tissue system, so it makes up a major part of the plant, the ground tissue. It is consisting of the parenchyma, collenchyma, and the sclerenchyma, about which you have already learnt. So this makes up the ground tissue system. In the stem, we can see this stem and the root. We can see the ground tissue in the form of cortex. So the cortex contains the ground tissue. Mostly it contains parenchyma. Sometimes it is also made up of collenchyma, and rarely it is also made up of sclerenchyma. So the cortex, the pith, the pericycle, all these parts along with the medullary rays present in between the two vascular bundles. We have medullary rays. All of this, it constitutes the ground tissue system. So it acts as a filler tissue, so providing support, mechanical strength, and being parenchymatous, they also help in storage of food and other waste substances. So that's about the second type of tissue, that is the ground tissue. And finally, we have the vascular tissue. So the vascular tissue system, as you know, it is made up of the xylem and the phloem. So the xylem and the phloem together, it makes up the vascular tissue system. As you know, the xylem and phloem are often present together, forming the vascular bundles. So they are present very close to each other. So we call what is known as the vascular bundles. Based on the presence or absence of cambium, the vascular bundles could be of two types. So if cambium is present, it is called as a open type of vascular bundle, and if the cambium is absent, then this type of vascular bundle is called as closed type of vascular bundle. So we have the xylem and phloem present together. So we have the xylem, and on the outer side we have the phloem. If the xylem and phloem are separated by a layer of cambium, then it is called as open type of vascular bundle. It is mostly seen in dicot stems, and it will result in the secondary growth of plants. Whereas in closed vascular bundle, which is mostly seen in grasses, that is in the monocots, the secondary growth is completely absent. So based on the presence of the cambium or absence of cambium, vascular bundles could be open or closed. Also, based on the position of xylem and phloem with each other, the vascular bundles are of three types. We have what is called as radial type of vascular bundle. We have the conjoint type of vascular bundle. Again, it could be either conjoint open or conjoint closed. In radial type of vascular bundle, what happens is we have the xylem and the phloem. They are present on alternate radius. So if this is a section we have taken, this is the xylem and this is the phloem. So they are present at different radius. Therefore, this type of vascular bundle is called as radial vascular bundle. Whereas in conjoint type of vascular bundle, conjoint present together. So in conjoint type of vascular bundle, what we see is both the xylem and phloem are present in the same radius. They are not present on different radius. They are present on the same radius. That's what is called as conjoint. If the xylem and the phloem, if they are separated by the cambium cell, cambium layer, it is called as conjoint open. If the cambium layer is absent, then it is called as conjoint closed. And always the phloem tissue is present outside to the xylem tissue. Xylem tissue is present inside, whereas phloem is present outside. That is about the vascular tissue system. So the outermost covering of plants is made up of the epidermal tissue system. The innermost parts we have the vascular tissue system. Everything other than the epidermal tissue system and the vascular tissue system is the ground tissue system. These are the different types of tissue systems that we see in plant roots, stem as well as in the leaves. When we talk about the vascular tissues, the xylem and the phloem, we have to talk about two conditions called as the exarch condition and the endarch condition. So, in the exarch condition, we have the protoxylem and the metaxylem. Their arrangement with respect to each other is important. What is protoxylem and metaxylem? Pro means first. So protoxylem is the xylem which that appears first. So that is the protoxylem. So during the initial growth of the plant, when the plant is young, when the stem and the roots are young, the first xylem that comes is the protoxylem. Metaxylem, it comes later on in the life. So metaxylem is formed later. So this is the later formed xylem. So in the exarch and the endarch condition, what happens is, in the exarch condition, the protoxylem is present towards the outside and the metaxylem is present towards the inside. So this is the protoxylem, this is the metaxylem. This kind of exarch condition is seen in the case of roots. So roots, they show exarch condition. When you take a section of a root, what you will see is, you will see the epidermal layer, let us say, and we can see cortex, we will see the endodermis layer and the pericycle layer, endodermis and pericycle, and we see the xylem and the phloem. So the xylem is present here and the phloem is present here in patches, the radial type of vascular bundle. Xylem and phloem are present on two different radiuses. So here in this case, the protoxylem is present towards the outside, whereas the metaxylem is present towards the inside. This condition is called as exarch condition, seen in the roots. The endarch condition is seen in the stem, wherein the protoxylem is present towards the inside and the later formed metaxylem is present towards the outside. This condition is called as end arc condition. So these are the two important terminologies we need to know. So stem, it could be dicot stem or monocot stem. They will have the end arc condition where the protoxylem is present inside, metaxylem is present towards the periphery. In the exarch condition seen in dicot and monocot roots, we have the metaxylem present towards the inside, that is towards the pith region, let us say, whereas the protoxylem which is formed first is present towards the periphery, called as the exarch condition. We don't find this in the leaf, exarch or end arc. Same thing with phloem also. In the case of phloem also, we have protophlloem and we have metaphloem. Phloem is produced first, whereas metaphloem is the phloem that is produced later. In protophloem, the sieve tubes present in the phloem tissue, sieve tubes are narrow, whereas in metaphloem, the sieve tubes, these are wider, the diameter is wide. So that is about the two important terminologies. Thank you.